This is ICSI 2001, Chapter 26, Part B. We will be finishing up the fifth theme for this lecture, which is the structure of DNA and RNA, and it'll be a relatively short lecture compared to the rest of them. We hit the other four topics last week, so we will just be covering the structure of DNA and RNA this week. The structure of DNA and RNA. Nucleic acids are complex and chemical structures that are the building blocks of nucleotides. Each nucleotide is made up of a sugar molecule that has 5 carbon atoms, which makes it 5-sided, a phosphate group, and a molecule containing nitrogen that is called a nitrogenous base. Alright, so here we have an RNA nucleotide and a DNA nucleotide. And there's one main thing that differentiates DNA from RNA, and it's the sugar. Alright, so with DNA, it's deoxyribonucleic acid, and with RNA, it's ribonucleic acid. So, with DNA, you are short one oxygen in the ribose, and in ribonucleic acid, you've got the extra oxygen in the sugar. And the sugar group, if you're wondering, is right here. So here's your sugar group. There's your sugar group. Your phosphate group is up here. And then your nitrogenous base is going to be up here. And so basically, whenever you're looking at these, your phosphate group will be the same. Your sugar, other than having the one less oxygen over here on the deoxyribonucleic side, will be the same. And then all of your nitrogenous bases will be the same except for one. We'll talk about that in just a second. DNA is the nucleic acid that functions as the original blueprint for making proteins. It contains the sugar deoxyribose. It contains phosphates, and the nitrogenous bases adenine, which we demark with an A, guanine, we demark with a G, cytosine with a C, and thymine with a T. A's will always double bond with a T, and C's will always triple bond with a G. So anytime you see a T, it'll be with an A, and a G will always be with a C. So just like that. A's will be with T's, G's will be with C's. And kind of the way that I say it is A's and T's are just kind of like dating, and then because they're double bonded, C's and G's are married, bonded for life, that kind of thing. But A's and T's, they just kind of date back and forth. And you'll see what I mean in just a second, because when we switch to RNA, those A's and T's are going to separate. Okay, but the C's and G's always stay together. So nitrogenous bases of DNA. Here you have your thymine. This is what it looks like here. Cytosine, adenine, and guanine. Now I'm not going to make you look at the structure of these on a test and be like, okay, now which one is this, ladies? Or, anything like that. I'm not going to make you do that. But what I am going to make you do is you're going to have to be able to differentiate between a pyrimidine and a purine. You're going to have to be able to tell me that thymine and cytosine are pyrimidines and that adenine and guanine are purines. And the easiest way to remember that is in DNA, your pyrimidines, let me get the eraser so that you can see this better, see the words. All right, when you have a pyrimidine, you see how there's a Y inside pyrimidine, and there's a Y in thymine and a Y in cytosine? Okay? There's a Y in pyrimidine, Y in thymine, Y in cytosine. Okay? That's the easiest way to remember their pyrimidines. It's just a memory thing. In purines, there's going to be a U in guanine, U in purine. There's not one in adenine, so you're just going to have to remember it. But in the pyrimidines, they have Y's in all of them, and there's no Y's in any of the purines. So if you can remember purines in DNA all have Y's, then you're good. But you will have to remember purines and pyrimidines. And also, remember that purines will bond with a pyrimidine. So adenine will bond with the thymine, guanine will bond with the cytosine. RNA is the nucleic acid that is directly involved in the making of proteins. So DNA is our blueprint. RNA is the one that makes the proteins. It contains the sugar ribose. Okay, so DNA contains deoxyribose. RNA contains ribose. It also contains phosphates and nitrogenous bases. Here's the big difference. Okay, it still contains adenine, which is A, guanine, which is G, cytosine, which is C. Here comes the big difference, though. Instead of containing thymine, it contains uracil, which is U. There is no thymine in RNA, and there's no uracil in DNA. Okay, they switch up. All right, so in your pyrimidines, remember how I was like, oh, there's a Y here. Uh-oh, there's not a Y here. You're just going to have to remember that thymine just switched out with uracil. Okay, so there is no thymine. There is no thymine anymore. It just switched the place with uracil. So, but uracil is a pyrimidine. You're going to have to know the difference between the pyrimidines and the purines in RNA, and you're just going to have to remember that thymine and uracil just switched places. So uracil took the place because uracil is going to bond with this adenine. So in RNA, A double bonds with U, U double bonds with A. And then here we have our little old couple that stayed together forever, cytosine and guanine. And they triple bonded. Okay? DNA and RNA also differ in another way in that DNA is a double molecule made up of two flexible strands that twist around each other in a coil or double helix resembling a twisted ladder. Okay, looks like this. This is your DNA. Okay, DNA. The four bases of the helix always pair in a definite way, which is your A bonds with T, and C, we couldn't really put that in there with your little bonds, but triple bonds with G. The paired bases are called complementary bases. So if I ask you on a test, what's the complementary base of adenine, you're going to tell me that the complementary base of adenine is thymine. If I ask you what the complementary base of cytosine, you're going to tell me that the complementary base of cytosine is going to be guanine. Here's the picture of DNA. You can see here you've got your deoxyribose sugar and your phosphate group. These make up your backbone of your DNA ladder, and then you've got your rungs that are actually made up of your nucleotide, and you can see how they nicely fit together. Okay, you can also see, if I erase this, you can also see how you've got double bonds here between the adenine and thymine, and you can see triple bonds between the cytosine and the guanine. And I'll erase that again so you can see that. Now, once again, DNA is your two-stranded double helix, whereas RNA is only single-stranded. Okay? So that is one difference. So if you're trying to keep up with some of the differences between DNA and RNA, you can kind of do it like this. If you have a DNA and RNA side, so that's the RNA side, this is the DNA side. You can say RNA has a single strand, DNA has a double strand. You can also say that RNA has the sugar ribose, where DNA has the sugar deoxyribose. And the last one is RNA has uracil as one of its pyrimidines, whereas DNA has thymine as one of its pyrimidines. Okay, so if you want a nice little list of three differences between DNA and RNA, those are three major differences between the two. So with our GPS standards with biodiversity and heredity, just finishing these up, we demonstrated the role of DNA and heredity today. Obviously, demonstrating the ability to explain the mechanisms for transmission of the traits between generations. We did that last week. And distinguish between inherited traits and learned behavior. We did that kind of last week. One thing that I want to talk about real quick, let's go back to the previous slide. I'm going to change colors, and I'm also going to erase this real quick. I want to talk about the importance of DNA versus RNA. We talked a little bit about the three differences between DNA and RNA, but we said that DNA was the blueprint for our cells and basically for our life, and how RNA is what made our proteins or coded for our proteins. But what's important is, and what's important about DNA having two strands versus RNA only having one strand, is it makes a change in the size of the DNA and RNA. Okay, if the DNA is double-stranded and the RNA is single-stranded, when we talked about the cell and we talked about in the nucleus there were nuclear pores. In those nuclear pores, the reason why we have nuclear pores is for something to be able to get out of the nucleus. And what's getting out of the nucleus is the RNA. So we don't want DNA to ever leave our nucleus. That would be bad because the DNA is our blueprint. We never want the blueprint to leave our nucleus. We got to have it there. But we have to be able to let something leave our nucleus to be able to give instructions to the rest of the cell. So that's what the RNA is there for. The RNA is what makes codes from the DNA to give the rest of the cell to make, basically it makes proteins. It helps, well, the ribosomes make the proteins, but it codes for the ribosomes to make the correct proteins to give to the rest of the cells to simplify the logistics of it. So that's the main differences between DNA and RNA, and there are different types of RNA that does this. There's messenger RNA, there's ribosomal RNA, there's transfer RNA, but basically it's all different types of RNA used during DNA transcription and translation and the replication. So just keep in mind that this is how those traits are passed on whenever we replicate that DNA during the synthesis part of our cell cycle that we talked about during our lecture a couple of weeks ago. When we're getting ready for the part in the cell cycle preparing for mitosis, for nuclear division, and we have to replicate our DNA. In order to replicate it properly, our RNA is what does that. So that's why it's important that these base pairs here line up properly, because if they don't, and it gives our RNA the incorrect code, then a mutation can happen. It can pass on the wrong traits. And when we're talking about inherited traits versus behavior traits, that would be something like an inherited trait would be, like your hair color or your eye color, your skin tone, your height, that kind of thing. And learned behavior would be knowing to brush your teeth, or learning how to read, or knowing how to act in a restaurant or in public, or manners, or learned behaviors. Instinct would be a mother bird knowing how to feed her young, or a mother cat knowing how to move her young or newborn cats without harming them. Those are instinctual things. Those are not learned things. The learned thing would be, the dog fetching the ball. That's learned, or the dog learning how to sit when commanded to do so. So, the inherited things are passed with these genes that's carried in the DNA versus being taught. Okay? So don't forget to participate in the discussion. This is due by next week, of course, and your quiz. And next week, you will be testing. Okay, so we have a test over all of chapter 20 and 26. So don't forget that. That is pretty important. So the next time we meet, we will have a test next week over chapters 20 and 26. So be prepared for that. And also don't forget, in the lab that we did with the genetics, with making the children, I said to make sure that you bring a drawing of the children that you created from your lab, and we're going to compare those. And because I believe there were some questions that we had to answer from that to finish up that lab. So don't forget to bring those, and I will see y'all at our next meeting time. Y'all have a great week.
Made with Doodlecast Pro.